Alrighty, welcome back. So we're going to continue with where we left off with acids and bases. So water disassociates into an equal number of hydrogen ions and hydroxide ions. So hydrogen ions are going to be used to hydrogen. They have a positive charge. And then hydroxide ions are going to be one oxygen and one hydrogen. This has a negative charge. So water, whenever it's exposed to just certain things, breaks down, and those are going to be the two products that we create. So for acidic solutions, they have a high hydrogen ion concentration. Lemon juice, vinegar, and coffee are some pretty common examples of this. These are all things that are going to be considered acidic. Acids release hydrogen ions or take up hydroxide ions. So hydrochloric acid is HCl. Whenever it's broken down, it turns into hydrogen ion and a chloride ion. So it's giving off a hydrogen ion, so it is considered an acid. The other side of that is that it takes up hydroxide ions. This isn't an example of that, but just know that that's a thing. Um, Basic solutions are going to be the opposite of that. So milk of magnesia and ammonia are going to be examples of this. Either take up hydrogen ions or release hydroxide ions. So it's going to be the exact opposite of the acid. So sodium hydroxide is NaOH, and that breaks down into a sodium ion, and it releases the hydroxide ion. So again, that hydroxide ion is being released, which is what causes it to be considered basic. Whenever we talk about acids and bases, we break it down into the pH scale. I'm sure you've heard about a pH scale. It's often talked about. A lot of people talk about it with their pool, like a chlorine. If you have any type of fish tank, you talk about pH within the fish tank because you want to make sure that you're not basically dissolving your fish. So the pH scale is going to be a mathematical way to indicate the number of hydrogen ions in a solution. So the pH scale ranges from 0 to 14. pH below 7 is considered acidic. It has, excuse me, it has more hydrogen ions than hydroxide ions. pH above 7 is considered basic. It has more hydroxide ions than hydrogen ions. And then a pH of 7 is neutral, and that's where both of those are just in equal parts. And there's a little graph showing kind of like common things that you may run into. So obviously hydrochloric acid going to be way up there in the acidity scale. Stuff like stomach acid is going to be right below it. Stuff that you encounter commonly, like lemon juice, Coca-Cola, tomatoes, black coffee, stuff like that. Those are still considered acidic, but you're getting a little bit lower down there. Then you have normal rainwater. So untainted rainwater is just water that's going to have, not a lot of stuff going on for it. Pure water and tears is going to be 7. Human blood is going to be slightly basic. You get seawater, baking soda, Great Salt Lake, milk of magnesia, so on and so forth. So just some kind of common stuff that you encounter that has certain levels of acidity or basicness. When we talk about that, we're also going to be talking about buffers. So buffers are going to be chemicals or combinations of chemicals that keep pH within normal limits. Resist pH change by taking up excess H or OH. pH of blood is about 7.35 to 7.45 and is maintained by a buffer. So literally all a buffer is something that we add to a mixture to uptake excess hydrogen or hydroxide ions to keep really close to neutral or wherever we need it to be. So if you have something that's way acidic and you don't need it to be that acidic, you add a buffer to it to lower that acidity by taking up those extra molecules. Same thing on the other side. If you have something that's too basic, you add a buffer to it and you can bring the acidity level up a little bit. So organic chemistry, the bane of my existence, this class I struggled with potentially the most. It was definitely a scary one. It's going to be the study of organic molecules. Organic molecules contain carbon and hydrogen, and inorganic molecules do not contain a combination of carbon and hydrogen. So there's an example of how these are written down there. The entire organic chemistry class involves writing chemicals out in this fashion. It is terrifying. It's a lot of work. It's really pretty. Like, don't get me wrong. Like the science behind it is beautiful. I'm just not good at it. But that's how it's written. It's very daunting to look at before you get into all of it. But you can kind of see a little bit of what we've talked about so far. So a lot of those lines there are just a single line, and we know that that's going to be a single bond. And then a lot of those lines have double lines, which means that that is going to be a double bond. So that's something that we've talked about that you can kind of see represented in an actual example of how things are written out whenever we talk about chemistry. So the carbon atom, one of the most important atoms in the world, or in potentially in the universe. Carbon has a total of 6 electrons, 4 of which are in the outer shell. Almost always shares electrons with elements such as hydrogen, nitrogen, and oxygen. Can bond with as many as 4 other elements. Most often shares electrons with other carbon atoms. Hydrocarbons are chains of carbon atoms bonded only to hydrogen atoms. And then isomers are going to be the same number and kinds of atoms in a variety of arrangements. And they may have different properties based on those arrangements. So carbon, as it says, has 4 electrons in its outermost shell. It's not the happiest, but it's not upset. It's really halfway. Like, carbon is just really chill dude. And he's able to share those electrons, and that's what he does most of the time. So if we think back to one of those early pictures I showed you during the last video, carbon was bonded with 2 oxygens, and that was able to hit those 8. So carbon is going to be like the backbone of like everything that we build off of. Carbon is super, super, super important. It doesn't have a lot of reactivity, which makes it really cool to work with. And it's able to bond with so many other things that it just becomes really stable and a really staple piece of organic chemistry. So there's a few examples of different ways things are written. Carbon chains can vary in length and have double bonds and or branch. So that's basically a carbon chain. It's just a chain of carbons all attached to each other. They'll have stuff coming off of offshoots that are written on them. But that's what they are. They're really helpful. They're what, like I said, the backbone of building things. And then carbon chains can form rings of different sizes and have double bonds. So if you'll notice in that situation, that first one has a lot more hydrogens. It actually has double the amount of hydrogens, but the second one has double bonds. So it got rid of the hydrogens, but is able to create double bonds with the carbon sitting next to it, which makes it also happy. But they make these rings, and these rings are really, really stable. A lot of things use the rings. So, oh God, what is it? Artificial sweeteners use them a lot. The one that starts with A that I'm completely blanking on right now. Aspartame. There we go. That one uses is called a benzene ring, and it is a ring of carbons like that. So they're pretty common. The carbon skeleton and functional groups. So carbon skeleton is the backbone of organic molecules, as I've just said. Functional groups are specific combination of bonded atoms that always have the same chemical properties and always react the same way. Reactivity of organic molecules largely dependent on attached functional groups. Often use R to stand for the remainder of the molecule. So that sounds a little bit confusing. But basically, if there's a part of an atom that is always the same, like no matter what it's on, it's always going to act the exact same way, you save time by just not writing it. It is an implied existence, and we put R to show that it's there. So some examples of it right here. We can replace different parts of writing this out with R. So for like that second example, the H3C, H3C is H3C. That's what it is. So in that situation, we can just get rid of it and write R, and it's called the functional group. It is implied that it is there. It exists. We just don't have to write it out every single time because it always acts the exact same way. and it's just faster. Scientists like doing things faster. So there are four categories of biological molecules: carbohydrates, lipids, proteins, and nucleic acids. Digestion breaks these molecules into subunits to build the macromolecules that make up the body. A lot of these you've probably heard about if you are at all into like dieting or I guess not dieting in general, but just taking care of your body in a very specific way. Some people will go really hard into eating specific types of macromolecules, and other people will like completely avoid other macromolecules. But it all goes into building your body. So if you're trying to build your body a certain way, you want more of some of them and less of others. So monomers are going to be subunits. Polymers are going to be monomers joined together. Dehydration synthesis reaction is joins monomers to form polymers, equivalent of removing a water molecule. So dehydration is removing water. That makes sense because if your body is dehydrated, you're obviously lacking water. So if you see anything that mentions a dehydration synthesis, that is bonding two monomers together to create a polymer, because again, polymers are just monomers joined together. So you're creating a polymer by just removing water and everything else is bonding together. Hydrolysis reaction is an OH group attaches to one monomer and H from water breaks to the other monomer, used to break bonds in a polymer. So this one is you are adding water to break whatever it is. So again, this is one of those where if you know the words, it makes sense. So hydro, again, is water. Lysis is to break. So this is using water to break apart a molecule, and that is exactly what it's called. So hydrolysis is adding water to a molecule to break it apart. So the first one we're going to talk about is going to be carbohydrates. So carbohydrates are mainly used for immediate energy source, maybe used for structural components. Carbohydrates are also classified as monosaccharides, disaccharides, and polysaccharides, which we will talk about as we go forward. But just as a little brief thing, saccharide is a type of sugar. So mono is one, you get that. Disaccharide is two, so it's two sugars. And then polysaccharide is three or more sugars all connected together. So there are some of the examples of common carbohydrates that you run into in daily life. Delicious. All right, so as I said, monosaccharide, I underlined mono in there because it is single. It is a single sugar molecule. These are simple sugars, 3 to 7 carbon backbone, and the most common one is going to be glucose. Your body produces glucose naturally. A lot of things in the environment produce glucose, and it is just a really simple sugar. It's easier for your body to make this than it is the other sugars. This is two isomers, fructose and gadacose. So cells use glucose as the energy source of choice. Ribose and deoxyribose are found in RNA and DNA, and these are sugars. So again, the most important part about monosaccharides, like the stuff that I want you to know about it, is that monosaccharide is a single sugar, and it is a simple sugar. Pretty simple. Going on, disaccharides, as I said, is going to be two monosaccharides bonded together. The common one for this is going to be maltose. So yeast breaks down maltose in beer for energy and produces ethyl alcohol, which is the fun stuff. This is also called fermentation. And this is also sucrose, which is table sugar, so something that's really, really common. But as you can see there, for the maltose, you have two glucose molecules that are bonded together through that oxygen bonding, and they create a disaccharide. There you go. There is the totally accurate representation of maltose being shoved into a beer can. So polysaccharides. Polysaccharides are used as the energy storage molecule. So the other ones were like the immediate storage. This one, or immediate energy use, this one is more of a storage system because it's harder to break. So poly is going to be three or more. These are going to be polymers of monosaccharides. So it's three or more monosaccharides that are all tied together. Some function as energy storage molecules. Plants store glucose as starch. Animals store glucose as glycogen. Some function as structural components, such as cellulose, which is the plant cell wall. Most abundant of all organic molecules. digested only by some microbes. And then chitin is going to be stuff like crab, lobster, insect exoskeletons. So polysaccharides can be really good for storing, or they can be used for structural components. And there's just some pictures of cellulose fibers with alternating bonds that make them a little bit stronger and harder to break. Very neat, I suppose. So the next one we're going to talk about are going to be lipids. So this is actually what I did part of my research on. So I'm pretty sure that I talked about it a little bit in my introduction video, but I worked on lipids that were stored inside of spiders. So lipids are going to be all insoluble in water, long nonpolar hydrocarbon chains, relative lack of hydrophilic functional groups, very diverse structures and functions, fats and oils used for long-term energy storage, and oil may help waterproof skin, hair, and feathers. So what I talked about with my spiders is that lipids are, as this says, a form of long-term energy storage. There's a whole bunch of it. So this says that there's very diverse structures and functions. So different lipids are going to be built differently, and how they are built depends on how they function. So some forms of lipids are meant for even longer storage than other ones. Some of them are really, really hard to break, which means they withstand stuff like heat. And so, like for my master's degree, I was looking at the lipids that are stored inside of spiders to see if, like, the ones that go around outside and they're in the daytime, they're exposed to high heat and stuff like that, if they had different types of energy storage than those that just like were only nocturnal and they just chilled on a web and didn't do much. So lipids have a whole bunch of different functions and different properties based on how they're built. There's a few examples that you common a lot in life. So cheese, ice cream, lard isn't as nearly as popular today as it used to be, but it's still there. And then butter and oil. Fats and oils are made up of molecules called triglycerides, which contain two subunit molecules. And those two subunit molecules are going to be glycerol and a fatty acid. So three carbon chain that has three OH groups is what's going to be a glycerol. And then fatty acids are going to be a long chain of carbon atoms bonded only to hydrogen with a carboxyl group at one end. So a triglyceride forms when the carboxyl portion of three fatty acids reacts with the OH groups of glycerol. This structure contains a lot of energy. Fats and oils are the body's primary long-term storage for energy. So what you want to know about this is basically these are hard to break. These are really long chains that are able to be folded and are able to be stored really, really well, and they're a little bit more difficult to break, which allows them to be good storage molecules. So here is a really quick picture of it, but you have that glycerol that has the OH group on the end of it, and then you have the three fatty acids. So those three fatty acids swing in, and they end up connecting to that OH group that was sticking off of the end of the glycerols. And that right there is what makes this the lipids. It's kind of neat, but I know the previous slide was kind of complicated sounding, so here's a nice little visual guide to kind of show it off. So continuing with fatty acids are the primary component of fats and oils. Unsaturated fats have double bonds in the carbon chain whenever the number of hydrogens is less than 2 per carbon atom. Trans fat is an unsaturated fat, one or more double/triple bonds between the molecules, and saturated fatty acids have no double bonds between carbon atoms. So unsaturated fats are going to have are going to have less hydrogen atoms connected to them, and they're going to have double bonds in there. Again, those double bonds are harder to break. And then the saturated fats, as you can see in that picture right there, they have all hydrogen atoms across it. They aren't rocking any double bonds except for the oxygen group that's over there on the side that we don't care about, because that's an R group. Look at that. But yeah, so we're mostly looking at the functional group, which is going to be that the rest of the atom. So phospholipids are going to be something that are found inside of a lot of things. They form the bulk of the plasma membrane, which is part of the cell. One end of the molecule is water soluble, and the other end of the molecule is not water soluble. So it has a polar phosphate head and a non-polar fatty acid tail. So how these are formed, over here you can see this red part is going to be the polar head. So this is water soluble, and then the inside of it, which I keep losing my mouse. There we go. This inside right here is going to be not water soluble. So it kind of repels water. It keeps it where it is, but it doesn't allow the water to easily transverse through the cell membrane. So it kind of keeps it protected. Steroids are going to be 4 fused rings. Steroids are lipids made of 4 rings. No, they have no fatty acids, but they are insoluble in water, which means they don't break down easily in water. They are derived from cholesterol and differ only in functional groups. So different steroids are going to be based on the functional group that's attached to them. But just know that steroids are lipids made of 4 fused rings. Proteins and nucleic acids. So proteins, many functional supports, many functions. They support metabolism, transport, defense, regulation, and motion. Proteins are comprised of amino acid monomers, central carbon bonded to a hydrogen atom amino group, carboxyl group, and a side chain or an R group. There are 20 different amino acids, and they differ according to the R group. So amino acids are going to be super, super, super important. They're basically what build us. They're what your body makes to turn everything inside of you into you. They are super important, and we get really in depth into them later on. If you ever have to take like the advanced biology classes, there's a good chance you'll have to know the 20 different amino acids and how they function. And that's terrifying. But just know that they are incredibly important, and they are the building blocks inside of you. So proteins are going to be pretty common. They're stuff that you see every day. So here are some eggs, some milk, some meat, tofu, nuts, and beans. Those are going to be super common ones. So whenever we talk about amino acids, got to talk about peptides. So peptides are 2 or more amino acids that are covalently linked. A peptide bond is formed by dehydration reaction between 2 amino acid monomers. So again, dehydration, you are removing water from the system, and that is creating a bond. A polypeptide is a chain of many amino acids joined by peptide bonding. So again, we're getting into the poly bonds. So poly is going to be 3 or more. We just have a whole bunch of amino acids that are all linked together. Amino acid sequences determine the final 3-dimensional shape of the protein. So these all have different shapes, and they all have different bonding. And as we've shown in that picture a couple slides ago with the lipids, the saturated versus unsaturated, whenever you had the double bond and you were lacking hydrogen, it was able to bend more, where if you had just all the carbon chain with the hydrogens on the outside, it was very, very straight. So there's a lot of shape that goes into so there's a lot of I guess what is inside of the molecule is what determines the shape that it is. And we use that a lot in chemistry and obviously a little bit in biology as well. So there's just an example of the synthesis and degradation of a peptide. You have the OH and the H, so those go together. You get a water that exits, and that creates the peptide bond through the dehydration reaction. You can also do the opposite of that and do a hydrolysis reaction where you add water to it, break down a dipeptide, and you get your 2 amino acids that now have the water broken down and attached to each side of it. So the shape of proteins. They are functional. The function of them is determined by the 3-dimensional shape that they are. Loss of structure and function is called denaturing. So you'll hear this a lot of times whenever you cook. This denatured, and it basically means that you heated it to such a degree that what it was started breaking down on you, usually due to pH or temperature. The primary structure is going to be amino acid sequences. The secondary structure is a portion of chain that forms helices or pleated sheets. So primary structure, again, amino acid sequence. So this is just amino acids that are connected to each other. Whenever you get a bunch of those that start going together, you get portions of chains that form helices or pleated sheets. So helices are going to be like a spiral shape. So if you think about DNA, DNA is called a double helix. So that's 2 helices that are twisted around each other. So just a helice is a spiral. Pleated sheets are going to be like whenever you fold your sheets, you just have layers upon layers upon layers of them all stacked on top of each other, nice and neat. Tertiary structure is going to be the overall three-dimensional shape of the interacting secondary structure. So this one is going to be looking at the shape of it all together. This is whenever it starts looking more 3D, which tertiary means three, so that kind of makes sense. And then quaternary structure is more than one polypeptide chain interacting. So you just get a whole bunch of mess of, I don't know, man, different colored spaghetti noodles that are all intertwined on top of each other. I got some pictures to make it a little bit easier, but this is going to be the primary structure. So as I said, this is all just kind of them linked together. This doesn't have a definite shape. This is just kind of like loosely thrown. Again, this is the equivalent of like a single noodle. Then you get secondary structure. This is whenever it starts taking shape. So you can see that this has a lot more bonds going on. It has a lot more double bonds going on. There's forced shape now because we've got a lot more chemicals interacting with each other. It's called an alpha helix. So just kind of know that secondary structure is whenever it starts taking shape, and it's got kind of like that loop going on for it. Then you get to tertiary. So tertiary is going to have two things. So that top one is going to be the pleated sheet, and you can kind of see why it's called that. It's pleated, which just means there's like a crease in it. So this one is kind of laid out. It still has a little bit of 3D to it. Sometimes they are laid out like this, where you just have like zigzag up and down kind of thing, but sometimes they are stacked. And then the other one is going to be a globular shape. So again, this is 3D, but this is just kind of like all of the proteins smashed on top of each other. And the last one is going to be the quaternary structure. So the blue, the red, and the green, each of those are going to be a respective individual polypeptide. But we take a bunch of them and we slam them together, and you get like a super duper 3D shape. But quaternary is just a bunch of things interacting with each other and kind of like sitting on top of each other. So nucleic acids, DNA, which is going to be deoxyribonucleic acid, or DNA, stores genetic information. So we've heard of this one. This is what makes your body. This is what tells your body how to function, how to build, how to do everything. And then we have ribonucleic acid, or RNA, and it helps make proteins. So there are polymers of nucleotide monomers. Nucleotides composed of a phosphate and 5-carbon sugar and a nitrogen-containing base. 5 types of bases, and that's going to be adenine, guanine, cytosine, and thymine. If you're in DNA, or if you have RNA, it's going to be uracil. So we're going to get to a point where we start like writing stuff out, and you can always tell if you're working with DNA because it's got the T, and if you're working with RNA, it's got the U. I think we talk about it here in a couple slides, maybe, yeah. But these always will match up with each other in very specific ways, so it's pretty easy to understand and write it. But there's an example of a nucleotide. So you have your sugar, you have the nitrogen-containing base attached onto one side of it, and then you have a phosphate attached to the other side. Fun. So specifically working with the DNA right now, DNA is going to be, again, the deoxyribose is going to be the main sugar, and this creates a double helix. So it works with complementary base pairing. Adenine is always going to be matched with thymine, and cytosine is always going to be matched with guanine. Genetic information is stored in the sequences of bases. So whenever you write these out, you'll usually see them written out as a form of just a bunch of letters written out. So we don't write out like adenine, thymine, adenine, 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 thymine. You write A, T, A, T, A, C, G, whatever. But you know what those stand for. And so it's a double helix, which means that there's pairing going on in between those two helices. And it's always going to be matched with its partner that's shown right here, A with T and C with G. And there you go. There's some beautiful examples of the two helices working together to form those the chains that structure it and hold them together and kind of force it to form that helix shape. So RNA, ribose is going to be the sugar this time. So again, sugar is going to be the main, the centerpiece of it. You got a phosphate on one side, you got a nitrogen-containing base on the other side. This is going to be single-stranded, and uracil is going to be the inside of it instead of T. So over here, you had T matching with A. In RNA, it's just going to be A matching with U. So the U completely replaces the T. And there you go. You don't have as much of a double strand going on. You just have the one strand, and then you have pieces attaching onto it. Comparing proteins and nucleic acids. So sequences of bases in DNA determine sequence of amino acids in a protein. Sequence of amino acids determine a protein's structure and function. Small changes in the DNA may cause large changes in a protein. And sickle cell anemia is going to be kind of an example of that. Individuals' red blood cells are sickle shaped, and there's one amino acid difference between that and a regular cell. And this is an inherited disease because we obviously inherit genetics from our parents. So if they have it, you get a chance of getting it. So we get more into this later on, but I'll touch on it real quick. But whenever you have a sequence of DNA, you have all those letters written out. There are things called insertions and deletions at their like super basic form. So insertion is where you take another amino acid and you jam it in there in the middle of something. So if you think about a word, if you think about a paragraph, like this slide right here, if I were just to jam a letter inside of one of those words, but keep the same number of letters in that word, that means that extra letter is going to be shifted over to the next word. And then that word is also the same number, so it's going to be shifted over one. And you end up with a whole bunch of new things that aren't technically words anymore. And so that's kind of how insertions work, is you get something inserted into your genome, and it reads in a base pair of three. So it's 1, 2, 3, stop, 1, 2, 3, stop. And that 1, 2, 3 is kind of coding. And so whenever you have something inserted into there, that 1, 2, 3 gets moved, and it's like 1, 2, 6 or whatever. And then the next one starts with that 3 that should have been on the end. So now it's 3, 1, 2. The next one is also going to be 3, 1, 2. So it shifts everything over a little bit. And it causes problems like that because each one of those is coding for something specific. And then deletion is going to be you delete one of them. So instead of 1, 2, 3, you get rid of that 2. So now it's 1, 3, but it needs to be read in a series of 3. So the one behind it, I suppose, would take that 1 and move it over. So now you have 1, 2, 1, and the next one is going to be 2, 3, 1. If that makes sense. It's a little bit confusing. It's a little bit esoteric, but we talk about it a little bit later on, and it kind of makes more sense. But here's an example of it a little bit. This one is just a swap. So I don't know if it's an insertion or a deletion that causes this, but basically whatever it codes for that specific protein is misread because of whatever, and it changes it, and whatever it changes it to completely changes the shape of your blood cell. Blood cells are very specifically built to capture the most amount of oxygen and stuff. And changing the shape like that drastically decreases the amount of oxygen that it can carry, which causes issues all over the place. All right, so there's your Winston, which means this slide has now ended. I'm going to talk a little bit right here at the end of it. So these first couple chapters are a little bit chaotic. I asked my in-person students so you guys are obviously an 8-week course. Well, I say that, but I don't know if I'm going to reuse these videos, in which case you may not be an 8-week course. Who knows? Ignore this if you're not an 8-week course. But anywho, so I talked to my in-person class and talked to them about these first couple chapters, and they said that this was basically a reiteration of high school. So a lot of things that we are going over right now are basically things that you should have learned in high school, and this is just a refresher. So that being said, these first couple of chapters are a little bit chaotic. We touch on a lot of different topics, and I kind of see it as like whenever you skip a rock on water, it hits the surface and then it moves to the next jump, and then it hits the surface and then jumps, hits the surface and jumps. And that's kind of what we're doing, and it feels really chaotic and not fun right now. But I just want to say, like, we do slow down. stuff does change up a little bit. So if you struggle with these first couple chapters, just rewatch the videos, reread my PowerPoints, kind of like do what you can to work through it. I'm always here if you have questions. So you can reach out to me via Canvas, or you can come in my office. That's super chill. But if it's overwhelming, just don't worry about it. Kind of do your best right now. Reach out to me if you need help, but just know that it calms down and it gets a lot more fun as we go along. But that's all I've got. So I will see you guys at the beginning of the next chapter.